Well, thank you for coming, everyone. Indeed, I did my PhD at the TUE in the group of orthopedic biomechanics. And today's talk will be about mimicking the human spine in the lab. So that's work that I did in the iPSpine project in the last couple of years. And it's Halloween, so I'm not in a costume, but this is the only image that I have today. The rest will not be spooky. Let's talk about the spine. So what you can see here in red in this skeleton is the spine of a human being, and it allows us to move in different movements, like you can see on this image. But it's actually not the bony part that allows us to move, but it's actually what's in between these vertebrae, the bony part. So the red thing here. What you can see here on the right side is the intervertebral disc. I'll just at my point as well. So in the center, you can see the intervertebral disc. Here we have the bony parts of the spine. This is the vertebrae, and this is the cartilage end plates here in yellow. They cover the top and the bottom, and they're important as a filter, but also to distribute loads evenly. On the outside of the intervertebral disc, we have various lamella of the annulus fibrosus. It's a highly ordered structure. And in the center, we have the nucleus pulposus. I was mainly busy with the nucleus pulposus during my PhD, and I will tell you why in a second. So the nucleus pulposus, if we were to zoom in and take a look at the matrix composition, we would see that there are some cells, nucleus pulposus cells in the adult human spine, nucleus pulposus. There's not a lot of them, but I'll tell you later why that's the case. We have some collagen, a very important molecule, but the most important molecule are so-called proteoglycans. And these proteoglycans are really important because they can attract and hold water. So much so that the water content in the intervertebral disc in the nucleus pulposus is approximately 80%. So you have to imagine that 80% of all the tissue in this nucleus pulposus is actually made up of water. That's quite a bit. And how does this work now, these proteoglycans, that they can attract so much water and hold it? It's quite comparable to diapers, actually. So in diapers, there are molecules added that are super absorbable, which means that they can take up a lot of water. And it's very similar to what happens in the nucleus pulposus. And when we now think about the function of the nucleus pulposus in the intervertebral disc, we can compare it to a bike tire. So when we take the coat of a bike tire and compare it to the annulus fibrosus, then we can take the tube and name it the nucleus pulposus. Now we assemble the bike tire, we take the tube, place it inside the coat, mount it on a wheel and inflate it. And now you can imagine that if there are forces acting on the wheel, that these forces are evenly distributed because there's pressurized air in the wheel. So actually, the pressurized air in the wheel is comparable to the water that is attracted to the nucleus pulposus. So this water that is attracted with these diaper-like molecules from these diaper-like molecules acts like the bike tire. This is the biomechanical function. And this is what I now explained. This is true for the healthy intervertebral disc. And let me guide you through this image. So in the center here, you see the nucleus pulposus. It's a cut open intervertebral disc. And on the side here, you have the annulus fibrosus. And here in the healthy intervertebral disc, it functions indeed like a bike tire. But what's the case? Why is that the reason? Because tissue is newly made and broken down at the same rate. So you can see that this is in a nice balance. So the cells in the nucleus pulposus, they make up new tissue and they break down tissue at the same rate. And this is where nothing is lost. But what happens over time, and this is a very early hallmark of degeneration, is that we lose some of these proteoglycans, the diaper-like molecules. This balance gets out of balance, so more tissue is being broken down than newly made. And this is comparable to someone actually reducing the air pressure in your tire. So what happens early is that we see a reduced glycosaminoglycan, reduced proteoglycan content, and we see some structural changes, and this is mild to moderate degeneration, or a bike tire with some air left, reduced air. Unfortunately, this is also progressive. So after some time, there's even more tissue broken down and even less tissue made. There's even less cells that can make tissue. And you can see that these changes are also progressive. So disc degeneration is progressive. And in the end, when we have severe degeneration, it is comparable to a flat tire. And this is, of course, a problem for people with back pain because it is invertible disc degeneration is highly associated with back pain. At the moment, we do not have a curative treatment that treats degeneration and thereby back pain. But there's also hope because researchers all around the world and we in the iPSpine consortium are investigating a method that you are all are aware of. So we want to regenerate the mild to moderately degenerated disc by injecting biomaterials in cells and then thereby make it healthy again, or at least stop the progression that I introduced earlier. This is comparable to a bike tire with some reduced air, and the slogan would be to pump up the tire before you get a flat tire. So a human disc would actually be the best model to study this because the title from today is the human spine in the lab. But the human invertible disc is very difficult to obtain due to obvious reasons. So we found a very good alternative, which is the cow tail disc. So we get the cow tails from the slaughterhouse, and then we clean it. So we remove all the surrounding tissue. And here you see the bony part. Here would be an invertible disc, and here would be an invertible disc in the cow tail. We cut open the invertible disc, and we take out, with a very sharp knife, we take out the nucleus pulposus. So here in the cut open disc, you can see on the outside the annulus fibrosus. Here is the annulus fibrosus. This is the nucleus pulposus. And here would be the cartilage end plates. You can see a thin layer of cartilage here. And why do we only take the nucleus pulposus and not the whole disc? Well, the nucleus pulposus is actually where disc degeneration is believed to start and where also regenerative therapies are focused on. So we can take one part of the disc and actually mimic a bigger situation, namely the degenerating invertible disc. And what happens if we now take this nucleus pulposus and we place it in a medium where there's a lot of nutrients? Well, it swells. Due to these proteoglycans that act like diapers, it will swell a lot, and actually the tissue will be destroyed over time. So we had to come up with a solution in the first place, where we developed a culture chamber. So we take the nucleus pulposus tissue, we place it between two platens, and this we place into the inner chamber. Then we close the inner chamber and place it into an outer chamber. This looks very difficult because it's a technical drawing, but what it actually is, it works like a matrushka, like the Russian doll. So we have a chamber, we place the chamber in the chamber, and the chamber again in the chamber. So that's how it works like. So the first thing that we could solve is to actually culture nucleus pulposus in a confined environment, like it would be in the invertible disc. The second question we had is, well, now we have healthy tissue from these cow tails, but we want to study invertible disc degeneration. So how do we do that? Well, we did a literature review of 129 studies that all took invertible discs or tissues of the invertible disc from animals, not from humans, and then it induced degeneration with there's a plethora of methods to induce degeneration that are actually all supposed to induce degeneration that is similar to human invertible disc degeneration. What we found is that this research field for speaking for science is still quite young. So it only started around 2000 with the first publications and was a bit quiet. And then in 2010, it started to take off. And actually, from 2013 onwards, we had around 10 publications per year. This means that inducing degeneration in invertible disc cultures is actually now an established method, and it's believed that it actually will continue like that. So here you can see all the different methods to induce degeneration, and there's really a lot of them. So we had to decide, how do we best mimic invertible disc degeneration with this knowledge? And we decided, well, there is this early hallmark that we already know. It's this loss of these proteoglycans, right? So like someone would reduce a little bit of air in your bike tire, and we found an enzyme called chondroitinase ABC, and that's actually works like a scissor. So it cuts proteoglycans and thereby reduces the proteoglycan content, like reduced air pressure. So we took this enzyme and we injected it into the nucleus pulposus of our culture. And what you can see here is the result of a culture with this enzyme. So this is the healthy tissue, and in blue you can see a staining of these proteoglycans. It's nicely distributed and it's a very nice blue. However, when we inject this enzyme, these proteoglycans are reduced, and you can see that there's less coloration and it's more disturbed. And we can also quantify that, so we can measure how much we lost. And when we did that, we saw that the glycosaminoglycans, this proteoglycan reduction, is actually comparable to mild to moderate degeneration. So now we have a model where we can induce mild to moderate degeneration using this enzyme. But there's another problem, or there's another focus of our group. Well, there is these compressive forces that act on the intervertebral disc, but these forces are actually dynamics. So when you're walking, the forces on your spine and on your intervertebral disc are dynamic. So what we did is we built a bioreactor. Bioreactor is a machine which can mimic the load of the intervertebral disc. So you can see here the springs, and they move up and down, which is a biomotor that we can control. And via this arm, it is connected to our culture chamber. And this culture chamber, this is this culture chamber here on the left, we have our nucleus pulposus tissue. So now it is compressed dynamically like it is in the human body. And what we can do is we can mimic a whole day of activity. So what you do usually during a day. Hopefully all of us sleep for around 8 hours. This is the night rest where the pressure is a little bit lower. And then we become active for 16 hours. And then we can mimic different activities of the day. We can repeat that for several days or even up to weeks. And thereby we can mimic the physiological loading. So the loading like it is actually in the human spine. So the next step that we added was dynamic loading. So we have now a very complex model already. And here, I would like to give you some background information for the next investigation that we did. So the intervertebral disc is the largest avascular organ of the human body, meaning that it does not have blood vessels. The blood vessels here in red are only on the outside. So here at the cartilage end plate, and here also at the outer annulus fibrosus. But here in the nucleus pulposus, there are no blood vessels. And this means that every nutrients that are transported via the blood have to be transported via this long distance. So everything via diffusion, it has to diffuse inside, meaning that in the center of the disc, there's a very harsh environment. So there's a lot of nutrients on the outside, that's the blood supply, but in the center, it's less. And it's the same for waste materials. So cells produce waste and it has to be transported out of the disc, but it's a very long distance, so it takes some time. And when we now would take a look at these, what this means for the cells in the nucleus pulposus, well, we see that there is very little sugar because that is transported via blood, oxygen as well, and it is quite acidic. That's the byproduct of one of the metabolites, one of the waste products that the cells produce. So it's a very, very harsh environment for these cells, and it's very difficult for cells to survive in the environment that is in the degenerated disc. We can mimic this by changing the parameters of the nutritious medium that we add to the culture. So we did that so we can mimic this now by this harsh environment that is found in the intervertebral disc, and we did this in combination with dynamic loading. So we had our nucleus pulposus tissue that was healthy. We dynamically loaded it, and we mimicked this very harsh microenvironment. And what we found is that the cell density, and so this is the cell density of the healthy cells, it is actually reduced when we dynamically load and have this harsh environment. And this was not surprising to us because we already knew, well, this is basically the harshest conditions a cell can have in the intervertebral disc. But we were interested in something else. So we counted the living cells. We can do this with a staining method. And we divided the nucleus pulposus tissue in top, core, and bottom. And on the right-hand side here, you see the living cells. These are the black dots. So all of the black dots that you see here, we can count them, and then we know how many living cells are in the tissue. Well, we did this for the center and for the periphery, so the top and the bottom. And in a healthy tissue, this is close to 1, meaning that there is as many living cells in the center as there is in the periphery. But when we now combined dynamic loading and the harsh microenvironment, we see that it is less than 1. It's approximately 0.5, 0.6. And this means that in the center, here in white, there's less cells than in the center, in the periphery. So that means that the cells died in the center, but not in the periphery, meaning that it is actually the distance that makes it so difficult for the nutrients to reach the center of the nucleus pulposus. But we also want to use this model to study regeneration, right? To pump up the tire again. And we did this using the previously developed model where we can inject, where we injected chondroitinase ABC, the enzyme that cuts away these proteoglycans to induce degeneration. Then we had cells, multicaudal progenitor cells, that we marked with the orange markers so that we can see them. We mixed it together with a hydrogel and with bioactive substance. That's NCM, if any one of you heard of it already. We mixed everything together, so the bioactive substance, the gel, and the cells, placed it into a syringe and injected it into our previously degenerated nucleus pulposus. And then we applied dynamic loading to this. So this might sound a bit much, but it's actually just degenerated nucleus pulposus, where we injected everything and then we dynamically loaded it and we cultured it for several weeks. And the research question was, well, these cells that we inject, can they actually survive this environment? And what you can see here in orange is surviving cells. So not only these cells are happy, but we were happy as well, because we could see that this is very promising. These cells can survive for several weeks in the dynamically loaded nucleus pulposus. And this is really important because it's a prerequisite for any treatment with using cells, that they survive at least. But what we could also see is that when we inject these cells together with this instructive matrix, with the hydrogel and the bioactive substance, that these cells are more healthy and more anabolic, meaning that they produce more of these proteoglycans than when we do not inject this. And this is very promising, but it's also there's more research necessary to prove and to show that this method is actually regenerating the nucleus pulposus. So to sum up what I investigated, well, first we developed a chamber to culture the nucleus pulposus in confinement in the laboratory. We then found an enzyme that can induce mild to moderate degeneration in these explants, in these ex vivo explants. We then built a bioreactor to dynamically load the nucleus pulposus, and finally we mimicked the harsh microenvironment by changing the constituents of the ingredients of our culture medium. And in this model can also be used, like you saw on the last slide, to study cell and biomaterial injections, like used for iPSpine. So in the future, we hopefully can investigate a method to pump up the tire again by using biomaterials and cells. And with this, I would like to point out one really important thing is that I did nothing else, that I did have help from a lot of people during this project. So from the TUE, from Utrecht University, from Nantes University in France, also in Galway, Sheffield, Switzerland, Davos, and Germany, University of Ulm. So there's a lot of people that are involved in such a big project. And with that, I would like to thank you for your attention, and I'm open for questions.